Good day, this is Professor Will, CBMD PhD. Today is Tuesday, June 11, 2019. It's 23:26, 52 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's get started here. Surprising enzymes found in giant ocean viruses. Findings could represent new drug targets for human pathogens. Date June 5, 2019. Source Woods Hole Oceanographic Institution. The new study furthers our knowledge of viruses in the sea and on land, and their potential to cause life-threatening illnesses. The findings, which examine newly identified genes carried by mysterious giant viruses, could represent potential new drug targets for giant viruses linked to human diseases. An international team of researchers searched more than 8,000 viruses' genomes and found that many newly discovered giant viruses contain multiple genes for a type of enzyme called cytochrome P450. P450 enzymes are common in animals, plants, and bacteria, but finding them in new viruses is unexpected. Prior to the giant viruses, it was never considered that viruses would have these genes. This is an extremely interesting finding. In animals, P450 enzymes metabolize drugs, make steroid hormones, and defend against pollutants. We have yet to find out what they are doing in these viruses, but for sure they are unique, unlike P450s in any other organism. P450 enzymes, which constitute one of the largest enzyme superfamilies known, may also have major implications for understanding chemical effects, both in the sea and in human disease processes. We know some giant viruses may be linked to some forms of pneumonia, so gaining a better understanding of them will help us to develop ways of tackling those viruses. The P450s could represent drug targets for giant viruses thought to contribute to some pneumonias. Finding P450 genes and enzymes in diverse viruses opens a new window on the evolution of these important enzymes, which may help in understanding the biology and the origin of the giant viruses themselves, which currently is unknown and hotly debated. Viruses are the most numerous biological entities on Earth, though giant viruses were not known until 2003, when a virus large enough to be seen, the light microscope, was discovered. More than 1,000 genes were identified in that first giant virus. By comparison, the influenza virus has 14 genes. Since then, additional giant viruses with many more genes and more P450s have been found worldwide, some with nearly 3,000 genes. Increasingly, giant viruses are being found in the oceans, including in the deep sea. Good day, this is Professor Will, CBMD PhD. Today is Tuesday, June 11, 2019, and it's 23:42, 12 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's get started here. Details of first historically recorded plaque pandemic revealed by ancient genomes. Date June 5, 2019. Source Max Planck Institute for the Science of Human History. Summary. Analysis of 8 new plaque genomes from the first plaque pandemic reveals previously unknown levels of plaque diversity and provides the first genetic evidence of the Justinianic plaque in the British Isles. The Justinianic plaque began in 541 in the Eastern Roman Empire, ruled at the time by the Emperor Justinian I, and recurrent outbreaks ravaged Europe and the Mediterranean basin for approximately 200 years. Contemporaneous records describe the extent of the pandemic, estimated to have wiped out up to 25% of the population of the Roman world at the time. Recent genetic studies revealed that the bacterium Yersinia pestis was the cause of the disease, but how it had spread and how the strains that appeared over the course of the pandemic were related to each other was previously unknown. In the current study, an international team of researchers led by the Max Planck Institute for the Science of Human History analyzed human remains from 21 sites with multiple burials in Australia, Britain, Germany, France, and Spain. They were able to reconstruct new Yersinia pestis genomes, allowing them to compare these strains to previously published ancient and modern genomes. Additionally, the team found the earliest genetic evidence of plaque in Britain from the Anglo-Saxon 6 of Edix Hill. By using a combination of archaeological dating and the position of this strain of Yersinia pestis in the evolutionary tree, the researchers concluded that the genome is likely related to an ambiguously described pestilence in the British Isles in 544 AD. High diversity of Yersinia pestis strains during the first pandemic. The researchers found that there was previously unknown diversity of strains of Yersinia pestis circulating in Europe between the 6th and 8th centuries AD. The 8 new genomes came from Britain, France, Germany, and Spain. The retrieval of genomes that span a wide geographic and temporal scope give us the opportunity to access Yersinia pestis microdiversity present in Europe during the first pandemic. The newly discovered genomes revealed that there were multiple closely related strains of Yersinia pestis circulating during the 200 years of first pandemic, some possibly at the same times and in the same regions. Despite the greatly increased number of genomes now available, the researchers were not able to clarify the onset of Yersinia plaque. The lineage likely emerged in Central Asia several hundred years ago before the first pandemic, but we interpret the current data as insufficient to resolve the origin of the Justinianic plaque as a human epidemic before it was first reported in Egypt in 541 AD. However, the fact that all genomes belong to the same lineage is indicative of a persistence of plaque in Europe or the Mediterranean basin over this time period, instead of multiple reintroductions. Possible evidence of convergent evolution is strains from two independent historical pandemics. Another interesting finding of the study was that plaques genomes appearing towards the end of the first pandemic showed a big deletion in their genetic code that included two virulence factors. Plaque genomes from the late stages of the second pandemic, some 800,000 years later, show a similar deletion covering the same region of the genomes. This is a possible example of convergent evolution, meaning that these Yersinia pestis strains independently evolve similar characteristics. Such changes may reflect an adaptation to a distinct ecological niche in Western Eurasia where the plaque was circulating during both pandemics. The current study offers new insights into the first historically documented plaque pandemic and provides additional clues alongside historical, archaeological, and paleoepidemiological evidence helping to answer outstanding questions. This study shows the potential of paleogenomic research for understanding historical and modern pandemics by comparing genomes across millennia. With more extensive sampling of possible plaque burials, we hope to contribute to the understanding of Yersinia pestis microevolution and its impact on humans during the course of past and present pandemics. Good day, this is Professor Will, CBMD, PhD, Hayes, Wednesday, June 12, 2019 at 0024, 30 seconds Eastern Standard Time. And this is a continuation of my scientific research. Let's get started here. A treasure map to understanding the epigenetic cause of disease. Date June 3, 2019. Source Baylor College of Medicine. Summary. Researchers have identified special regions of the genome where a blood sample can be used to infer epigenetic regulations throughout the body, allowing scientists to test for epigenetic causes of diseases. Epigenetics is a system for molecular marking of DNA. It tells the different cells in the body which genes to turn on or off in that cell type. But the cell-specific nature of epigenetics makes it challenging to study. Whereas a blood sample can be used to genotype an individual, most epigenetic marks in blood DNA provide no clues about epigenetic dysregulation in other parts of the body, such as the brain or heart. Research teams identify special regions of the genome where a blood sample can be used to infer epigenetic regulations throughout the body, allowing scientists to test for epigenetic causes of disease. To do this, they focused on the most stable form of epigenetic regulation, DNA methylation. This addition of methyl groups to the DNA molecule occurs in the embryonic state and can impact health for your entire life. To identify genome regions in which DNA methylation differs between people but is consistent across different tissues, they profile DNA methylation throughout the genome in 3 tissues, thyroid, heart, and brain, from each of 10 cadavers. Since these tissues each represent a different layer of the early embryo, we are essentially going back in time to events that occurred during early embryonic development. To map DNA methylation, we converted methylation information into a genetic signal, then sequenced the genome. Our atlas required massive amounts of sequencing data, 370 times more than were used for the first map for the human genome in 2001. The nearly 10,000 regions the researchers mapped out, called correlated regions of systematic inter-individual variation, comprise a previously unrecognized level of molecular individuality in humans. Recent studies are already showing that methylation at these regions is associated with a range of human diseases, including obesity, cancer, autism, Alzheimer's disease, and cleft palate. Because epigenetic marking has the power to stably silence or stably activate genes, any disease that has a genetic basis could equally likely have an epigenetic basis. There is incredible potential for us to understand disease processes from an epigenetic perspective. CORSIVs are the entryway to that. Good day, this is Professor Will, CBMD, PhD. Today is Wednesday, June 12, 2019 at 0046, 8 seconds Eastern Standard Time. And this is a continuation of my scientific research, so let's get started here. Sleeping with artificial light at night associated with weight gain in women. Date June 10, 2019. Summary. Sleeping with the television or light on in the room may be at risk factor for gaining weight or developing obesity, according to scientists. The research is the first to find an association between sleeping with artificial light at night and weight gain in women. The results suggest that cutting off lights at bedtime could reduce women's chances of becoming obese. Although poor sleep by itself was associated with obesity and weight gain, it did not explain the associations between exposure to artificial light while sleeping and weight. Humans are genetically adapted to a natural environment consisting of sunlight during the day and darkness at night. Exposure to artificial light at night may alter hormones and other biological processes in ways that raise the risk of health conditions like obesity. Unhealthy high-calorie diet and sedentary behaviors have been the most commonly cited factors to explain the continuing rise in obesity. This study highlights the importance of artificial light at night and gives women who sleep with lights or the television on a way to improve their health. Good day, this is Professor Will, CBMD PhD. Today's Wednesday, June 12, 2019, 00:53, 1 second Eastern Standard Time. This is the end of this module on scientific research. Hope you enjoyed listening. Have a great day.